My name is Dr. Shabnam Haghighat Khajavi. I am an assistant professor in Islamic Azad University in Tehran, Iran. I am thankful to Dr. Waqas Bernay and California Research Institute for giving me the opportunity to participate in this symposium. Today, I am going to talk about role of whey protein concentrate and spearmint essential oil coating on the oxidation and microbial activity in cold storage of minced meat. In this study, application of spearmint essential oil as a great source of polyphenolic compounds such as carvone, cis-carveol, and limonene with antimicrobial and antioxidant activities incorporated with whey protein biodegradable edible base coating were examined in order to increase the shelf life and reduce the lipid oxidation of minced common kill cow meatballs. In this study, application of spearmint essential oil as a great source of phenolic compounds such as carvone, cis-carveol, and limonene with antimicrobial and antioxidant activities incorporated with whey protein biodegradable edible-based coating were examined in order to increase the shelf life and reduce the lipid oxidation of minced common kill cow meatballs during refrigerated storage. Whey protein concentrate as an edible coating has been used due to its high nutritional value as well as excellent oxygen and aroma barrier properties. Microbial and chemical experiments have been performed in order to evaluate the antimicrobial and antioxidant effects of this coating as natural preservative. All samples were tested against gram-positive Staphylococcus aureus and gram-negative Pseudomonas fluorescens, and Escherichia coli recognized as important food pathogens and spoilage bacteria in fish. Methods applied in order to prepare spearmint essential oil, as well as preparing the minced fish meatballs, are presented, and I will briefly talk about them. In order to prepare spearmint essential oil, aerial part of the plant were air dried at ambient temperature and were ground before extraction. The essential oil from 150 grams of plant was obtained by hydrodistillation for 3 hours using a Clevenger type apparatus at atmospheric temperature. The extracted essential oil was dehydrated by anhydrous sodium sulfate and kept airtight in dark at 4 degrees Celsius under nitrogen gas atmosphere. In order to prepare fish samples, the fresh fish were transferred in ice to the laboratory, were washed, and their skin, head, and bone were separated, and then they were filleted and washed. The fillets were minced and were divided into 50-gram rounded balls. 2 concentrations, including 0.5 and 1% weight per volume of spearmint essential oil were prepared. For preparation of whey protein-based coating solution, 5% weight per volume of whey protein concentrate solution was dissolved in distilled water under agitation for 20 minutes at an ambient temperature. Glycerol was used as a plasticizer, and the solution was incubated in water bath at 90 degrees for 20 minutes. The solution was then cooled to ambient temperature, and the spearmint essential oil was added at different concentrations, 0.1, 2, 3, 5, and 1% weight per volume, and homogenized for 3 minutes. Samples containing 50 grams of minced fish meatballs were assigned into control group, which means without using the coating, treated with 0.5% weight per volume of spearmint essential oil, treated with 1% weight per volume of spearmint essential oil, treated with only whey protein coating, treated with whey protein coating plus 0.5% weight per volume of spearmint essential oil, and finally treated with whey protein concentrate plus 1% weight per volume of spearmint essential oil. In order to prepare coated samples, the fish meatballs were dipped in different coating solutions as described for 2 minutes, and also they were allowed to drain. All the samples were prepared in triplicate, were packed in polyethylene airtight bags and stored in 4 degrees Celsius in fridge for 20 days. In order to determine microbial loads of the minced fish meat during storage, 25 grams of each treated samples transferred into sterile sampling bags, diluted with 225 milliliter of sterilized peptone water, and then homogenized at 230 rounds per minute for 2 minutes. Homogenized samples were then serial diluted. Table 1 represents the antioxidant activity of spearmint essential oil carried out by DPPH assay. According to Table 1, the increase in the antioxidant activity was concentrate dependent, and the spearmint essential oil exhibited considerable free radical scavenging activity with IC50 value of 0.44% weight per volume. The considerable antioxidant activity might be related to the existence of different terpenoids and the major phenolic compounds in the plant, especially carbone, cis-carveol, and limonene spearmint essential oil. The first part of the experiment was microbial analysis, so I will follow up first with this section. Figure 1 represents the changes in total viable count during the refrigerated storage. According to Figure 1, TVC of all samples was gradually increased during the storage period. Based on these results, samples treated with spearmint essential oil and those treated with whey protein concentrate without spearmint essential oil maintained quality of the minced fish samples up to day 11. However, quality of whey protein concentrate plus spearmint essential oil coatings treated samples maintained acceptable by day 10 of storage in fridge. By the end of the storage period, the maximum level of inhibition was observed for whey protein concentrate plus 1% weight per volume spearmint essential oil coated sample. Figure 2 shows the psychotropic bacteria. Figure 2 shows the psychotropic bacteria count (PTC) during the refrigerated storage. According to the obtained results, growing trends of the psychotropic bacteria count showed an increased pattern during 20 days of storage. As seen, growth rate of psychotropic bacteria in the treated samples was lower than the control sample. The obtained results showed that the most effective treatment for PTC growth inhibition was whey protein concentrate plus 1% weight per volume spearmint essential oil during the storage period. Figure 3 represents the change in lactic acid bacteria, LAB, during the refrigerated storage. Lactic acid bacteria, as gram-positive bacteria, can produce lactic acid by fermentation metabolism and also are among healthy fish natural microflora. They are among harmless bacteria in producing strains which may prevent growth of other harmful microorganisms. According to the obtained results, the whey protein plus spearmint essential oil treatments significantly lowered population of lactic acid bacteria in the samples compared to the control by the day 20 of the storage period. Among all the treatments, the whey protein concentrate plus 1% weight per volume of spearmint essential oil treatment had the highest ability in limiting the LAB growth. The second part of the experiments belongs to the chemical analysis. As shown in Figure 4, the changes of the samples during the refrigerated storage. Second part of the experiments belongs to the chemical analysis. Figure 4 shows the pH changes of the samples during the refrigerated storage. The pH is one of the most important indicators of fish freshness. Changes in pH value might be related to production of metabolites from the bacteria, including basic nitrogen and lactic acid during bacterial growth. In addition, activity of bacterial enzymes may lead to destruction of amino acids and also an increase in the amount of free fatty acids, which can influence pH of the sample during storage. It was observed that pH of the whey protein concentrate plus 1% weight per volume of spearmint essential oil treated samples was 6.9 and was increased to 7 or 7.2 by the day 15 and then was decreased to 7 by the day 20. This drop could be due to the formation of metabolites such as organic acids by lactic acid bacteria. Peroxide value of the samples measured during the refrigerated storage are presented in Figure 5. Rancidity in fish and seafood can be monitored by determining the amount of peroxide value and formation of malondialdehyde. Therefore, PV is one of the most important and effective parameters to investigate quality of food products with high fat by measuring the primary products of oxidative degradation. Results showed that peroxide value of all samples was increased significantly with time throughout storage period. The results indicated that whey protein-based coating enriched with 1% weight per volume of spearmint essential oil was effective in retarding production of peroxide in the minced homunculus stored in refrigerator, which might be explained by antioxidant and oxygen barrier action of whey protein concentrate incorporated with spearmint essential oil and inhibiting lipid oxidation. Figure 6 shows the changes in Q-barbituric acid during the refrigerated storage of minced meat. TBA, as one of the methods commonly used to determine concentration of malondialdehyde, is also known as the secondary product of lipid oxidation caused by endogenous enzymes of spoilage bacteria and indicates the fish deterioration. As shown, TBA value were 0.58. As shown, TBA values were 0.58 milligram malondialdehyde per kilogram for all samples in the beginning of the experiment. Afterward, increasing trend of TBA value in all samples has higher pattern or no significant changes during refrigerated storage observed. This increase indicated oxidation of lipids and formation of malondialdehydes as the secondary oxidation products. However, no decrease was observed in TBA values during storage period, indicating that breakdown of malondialdehyde to tertiary lipid oxidation products had not occurred. Figure 7 represents changes in total volatile basic nitrogen TVBN during the refrigerated storage. Total volatile basic nitrogen is a physicochemical index used to investigate quality of seafood and fishery products. The initial TVBN value for the sample ranged from 9.83 to 9.98 milligram per 100 grams of whey protein concentrate plus 0.5% weight per volume of spearmint essential oil treated samples and 1% weight per volume essential oil treated samples, respectively. From day 5, TVBN concentrations were significantly increased due to the formation of ammonia, trimethylamine and dimethylamine in all samples during storage as a result of activities of spoilage bacteria and endogenous enzymes. This increase was significantly higher for control sample than the treated sample. According to these results, quality of the treated samples remained acceptable more than the control sample, which was due to the effect of spearmint essential oil in combination with whey protein concentrate in reducing activity of spoilage bacteria and endogenous enzymes. Finally, we reached the discussion. The considerable antioxidant activity might be related to the existence of different terpenoids and the major phenolic compounds in mentha spicata or spearmint, especially carvone, cis-carveol, and limonene. And finally, we reached the discussion part. The considerable antioxidant activity might be related to the existence of different terpenoids and the major phenolic compounds in spearmint plant, especially carvone, cis-carveol, and limonene. And finally, we reached the discussion part. The considerable antioxidant activity might be related to the existence of different terpenoids and the major phenolic compounds in mentha spicata plant, especially carvone, cis-carveol, and limonene in spearmint essential oil. Whey protein concentrate-based coatings. Whey protein concentrate-based coating enriched with spearmint essential oil can be considered as a potentially effective method to prolong shelf life and increase quality of minced fish samples during the storage, and can act as an oxygen barrier compound and inhibit oxidation of fatty acids, especially polyunsaturated fatty acids in fish. Among all the treatments, the whey protein concentrate plus 1% weight per volume spearmint essential oil treatment had the highest ability in limiting the lactic acid bacterial growth. Accordingly, growth rate of lactic acid bacteria was decreased in the treated samples, which is due to the application of spearmint essential oil incorporated with whey protein-based coatings and the treated sample. pH of whey protein concentrate plus 1% of weight per volume spearmint essential oil treated samples were significantly lower compared to the control sample, indicating that incorporation of whey protein concentrate with higher concentrations of spearmint essential oil to coating significantly inhibited microbial count and reduced formation of alkali compounds such as ammonia and trimethylamine, and prolonged shelf life of the minced flesh of common kilka. Trend of changes in PV and TBA values for the minced common kilka treated with WPC plus 1% weight per volume of spearmint essential oil indicated that the primary and secondary decomposition were substantially inhibited. The results also indicated that whey protein concentrate-based coating enriched with 1% weight per volume spearmint essential oil was effective in retarding production of peroxide in the minced common kilka stored in refrigerator, which might be explained by oxidation and oxygen barrier action of whey protein coating incorporated with spearmint essential oil. This coating inhibited lipid oxidation by this oxygen barrier action. The spearmint essential oil incorporated with whey protein concentrate-based coating exhibited both antioxidant and antimicrobial activities. The antimicrobial and antioxidant activities has been controlled based on the phenolic compounds exist in spearmint essential oil. The antimicrobial and antioxidant effects of phenolic compounds in spearmint essential oil are contributed to destruction of cell wall lipopolysaccharides in gram-positive. The antimicrobial and antioxidant effects of polyphenolic compounds in spearmint essential oil are contributed to destruction. The antimicrobial and antioxidant effects of phenolic compounds in spearmint essential oil are contributed to destruction of cell wall lipopolysaccharides in gram-negative bacteria and the increase in membrane permeability for both gram-positive and gram-negative tested foodborne pathogens and spoilage strains, which influenced their cell metabolism and resulted in death. Accordingly, whey protein-based incorporated with spearmint essential oil reduced and inhibited activity of the studied bacterial strains and led to delayed information of TVB. This inhibition could be due to the hydrophobicity of the essential oil and its polyphenols penetrating lipids. Accordingly, whey protein-based coating incorporated with spearmint essential oil reduced and inhibited activity of the studied bacterial strains and led to delay information of TVB. This inhibition could be due to hydrophobicity of the essential oil and its polyphenols penetrating lipids of the bacterial cell membrane, damaging cytoplasmic membrane. Accordingly, whey protein-based coating incorporated with spearmint essential oil reduced and inhibited activity of the studied bacterial strains and led to delay information of TVB. This inhibition could be due to hydrophobicity of the essential oil and its polyphenols penetrating to the bacterial cell membrane, damaging cytoplasm. Accordingly, whey protein-based coating incorporated with spearmint essential oil. Accordingly, whey protein-based coating incorporated with spearmint essential oil reduced and inhibited activity of the studied bacterial strains and led to delay information of TVB. This inhibition could be due to the hydrophobicity of the essential oil. The antimicrobial and antioxidant effects of polyphenolic compounds in spearmint essential oil are attributed to destruction of cell wall lipopolysaccharides in gram-negative bacteria and the increase in membrane permeability for both gram-positive and gram-negative tested foodborne pathogens and spoilage strains, which influenced their cell metabolism and resulted in death. Accordingly, whey protein-based coating incorporated with spearmint essential oil reduced and inhibited activity of the studied bacterial strains and led to delay information of TVB. This inhibition could be due to hydrophobicity of the spearmint essential oil and its polyphenolic penetrating lipids of the bacterial cell membrane, damaging cytoplasmic membrane permeability, disrupting membrane transport function, interfering with generation of cellular adenosine triphosphate, ATP, and disrupting proton motive force, which in turn inhibits growth and results in the decrease in the bacterial count and death. And finally, the conclusion part, the use of spearmint essential oil. Finally, in the conclusion, we can describe that the use of essential oils as natural antimicrobial and antioxidant agents incorporated in coatings and films has received a great deal of interest in the field of food preservation. Therefore, the use of essential oils as natural antimicrobial and antioxidant agents incorporated in coatings and films has received a great deal of interest in the field of food preservation. Therefore, in the current research, edible coating was developed by incorporation of carbon-rich spearmint essential oil with whey protein concentrate solution. According to our results, the oxygenated compounds and polyphenols of spearmint essential oil at higher concentration of 1% weight per volume incorporated with whey protein-based coating as an oxygen barrier matrix that helps to slow release of essential oil showed significant slowing down of microbial growth by their lag phase extension and reduction of growth rate, and led to retardation or inhibition of growth of Pseudomonas fluorescens, Escherichia coli, and Staphylococcus aureus as representatives of gram-negative and gram-positive pathogenic and spoilage bacteria. According to the results, whey protein concentrate plus 1% spearmint essential oil treatment delayed or even prevented both growth of microorganisms, and formation of total volatile nitrogen, and pH changes without any effect on lipid oxidation by reducing PV and TBA, and represented a considerable antioxidant effect, decreasing enzymatic spoilage and preventing deterioration in the minced kilka during cold storage compared to control and WPC-free essential oil samples. Therefore, the edible coating could provide an extended shelf life, safety, and quality by better performance to inactivate post-processing contaminations on cold stored ready-to-eat foods such as the minced kilka meat, and could be potentially used as a promising natural food preservative. Thank you very much for your attention. Have a great time.